Good morning, good afternoon, good evening to all of you, wherever you are. And I am myself, Irfan Rahman from the University of Rochester Medical Center, upstate New York. And this is a working group retreat, which is lung aging research group from the Assembly on Respiratory Cell Molecular Biology. And we have been doing this quite a few years and been very successful. And we are a little bit delayed in terms of this arranging this retreat. However, we are here. So, as I said, my name is Irfan Rahman. Then I would like to introduce my co-chairs, Beata Kosmider. Beata, please go ahead.
Good afternoon. My name is Beata Kosmider. I'm a professor and director of the Center for Inflammation and Lung Research at Temple University in Philadelphia.
Mareike, please introduce.
Good evening for me from my side. My name is Mareike Lehmann. I'm a professor at the University of Marburg in Germany, so in a different time zone. And I'm interested in developing human models for lung aging. I'm looking forward to this session.
Correct. Thank you. So all of you, please, if you want to take a screenshot of this or save this email IDs, because these email IDs you can communicate to us directly, and you can provide feedback, suggestions, and any future retreat, seminar, webinar, or any suggestions. Or you can reach out to the assembly on RCMB or the chair of the assembly. So I'll give it 10 seconds if you'd like to take a screenshot or save this slide. Okay, so our key objectives for this working group are increase the awareness and disseminate the knowledge and basic biology of lung aging, understand the cellular molecular mechanism of aging and age-related diseases. For example, we have pulmonary fibrosis, COPD, and so other diseases. So develop therapeutics targeting age-related dysfunction spanning developmental to late life. Use of emerging technologies for cellular mapping of lung aging. So this is again, these are the key objectives, but can be more. And also we do have another group, which is critical care group, which also does a lot of age-related clinical research and also very similar to what we have here. So we collaborate with them and we are part of the ATS. So just please be mindful that there are multiple groups, but doesn't mean that you cannot reach out to us or them or both of us. So today is a special day. We have 90 minutes and we are 12 noon Eastern time until 1:30 p.m. Eastern time. So you will hear two presentations today. One is by Dr. Aditi Gurkar and second one is Dr. Hongwei Yao. And so we have divided into 30 minutes and another 30 minutes, but in between that we'll take some few questions. And then there will be a question answer. You can put your questions or suggestions on chat or reach out to us. And there will be panelists that will include speakers and member of executive committee of lung aging group. So this is pretty much what is today's retreat will be. And I would like to ask Dr. Beata Kosmider, my colleague, my co-chair, to introduce Dr. Aditi Gurkar. Please go ahead.
Thank you for joining us today. I would like to introduce our first speaker, Dr. Aditi Gurkar. She is an assistant professor in the Aging Institute at the University of Pittsburgh, and her research interests are focused on understanding the biology of aging and age-related diseases. Dr. Gurkar studies the signaling mechanisms that drive aging, which can lead to identifying novel therapeutic targets to extend the health span. So, Dr. Gurkar, we are very excited to have you here, and you may now begin your presentation.
Thank you very much. Thank you to the organizing committee for inviting me here today. Disclaimer is that my lab actually does not work a lot on lung biology. And so when I was asked to come here, I was super excited because, and I'll get into the reasons why, but my lab's been primarily interested in studying aging and particularly the biology of aging. And I hope today I can convince you that aging biology can be applied to the lung and hopefully learn lessons from you about the lung and apply it to my science. With that in mind, I always believe that science communication starts at home. And so when I told my daughter about the presentation today, this is what she drew for me. So I always try to include something that my kids do so that they can kind of learn more about science. All right, so why is my lab particularly interested in aging? Since we are all aging every minute of the day, I will get right into it. But if you look around, 1 in 6 Americans is about the age of 65. And our elderly population that is about the age of 65, will nearly double by 2040. And reach to about 80 million people in the US alone. And truly, are we ready for this gray tsunami? Now, most of you have already heard that age is the primary risk factor for many diseases. So when you think about cancers, when you think about diabetes, when you think about cardiovascular disease, and we'll talk about lung disease as well. But in all of these cases, the incidence of all of these go up significantly with age. In fact, if you look at the population about the age of 60 or so, about 45% of this population suffers from comorbidities. That's not just one disease at a time, but multiple diseases at one time. And what this translates into is a substantial cost, as well as not only quality of life that is quite bad for these elderly, but also quality of life for their caretakers, children, and our healthcare system in general. In fact, it is thought that if we can shift this period of comorbidities and improve health span by just about 2 years, we can probably save about $7.1 trillion by 2050. In multiple programs like healthcare, et cetera. And this is a substantial number. And so when we think about aging, most of us think about, frailty, loss of function, and disease. And this is the picture that comes to most of our minds. And in the field of aging, we think about the geroscience hypothesis. And what this is instead of studying one disease at a time, we think we could study the biology of aging in general. And if you can target the biology of aging in itself, we think this would improve or delay the incidence of all of these diseases collectively. And so this would improve health and health span. And so that's kind of the idea of geroscience and the biology of aging. Now, I was invited here to, talk and share my science with lung aging enthusiasts. And so I put in, I was starting to read a lot more about this. And obviously, like other chronic diseases, with lung aging comes a number of diseases, including COPD, where, again, the incidence of COPD increases with age. And so goes for multiple respiratory infections like COVID, right? We just went through this, and in most cases, 8 out of the 10 reported deaths in the US were for people over the age of 65. And although we've talked about disease and age being the primary risk factor, I always like to think about exceptions. In general, I think I'm a fairly optimist person, so I always like to look at the bright side. And so let's talk about exceptions for a little bit. And so when you think about aging and you think about functional loss and disease, there is also another side of this coin. And there are people who seem to be doing extraordinary things at their age, right? My favorite is really this gymnast, Joanna Quas, who is now, I think, 97. I think this was an older slide that I had. And she still traveled the world. But not only is this picture for me so impressive because she, is obviously in the best health ever, but the awe on these kids' face is amazing to see. Like, oh yeah, like I wish my grandma could do that, right? So clearly, age just is a number, and different people, even though they chronologically may be the same age, biologically, they definitely seem to be doing better and functionally fit. So when we think about biological age, I think trying to get the distinction between biological age and chronological age is very important. So when most of us, again, think about aging, we actually think about, comorbidities. So when one person is, or the elderly is hit with one morbidity, soon enough, 5 to 10 years later, usually other comorbidities hit in. And so the quality of life is really morbid. Versus, and in this case, one's biological age is accelerated compared to their chronological age. What most of us would really like is to see what's on the left side of this image, where wish we could have an amazing health span. And then maybe, yes, it's impossible to, probably not have any disease. But then we do get hit by that disease. And then a few years later, death. And in that case, we are functional almost till the end of life. And in this case, we think of our biological age to be very similar to our chronological age. So my lab's really been interested in these three sort of major questions. What is one's biological age compared to their chronological age? Can we define a molecular fingerprint for biological age? And can we modify biological age? Now, as you can imagine, this is a very complex question because unlike chronological age, which is on your license or on your passport, biological age to capture is going to be challenging. But we think we want to really try to do this because the implications of figuring out what one's biological age is, and if we can do this early on, we could really improve health and, reduce the burden on our healthcare system. And what was interesting while I was reading about lung aging was, and this is a very interesting paper, if you've not had the chance or opportunity to look at this, probably you should. But in this paper, they looked at organ aging. So they looked at multiple organs at a time and looked at aging of these organs through multiple methods, including function, etc. And what was interesting to me was that lung function feeds into function of most other organs. Maybe as lung enthusiasts, all of you already believed in it and thought lung was at the top of this. But I was very, very interested to see this data where pulmonary function was really kind of dictating the aging of other tissues. And also the hazard ratio, again, morbidity in these people, if they had better pulmonary function, they seemed to be much healthier, did not suffer from frailty, did not suffer from rehospitalization, etc. So it does seem like pulmonary function feeds into biological age quite a bit. So I got really excited about this and, hope to use this in all our future studies now. But how do we start thinking about how can we decipher someone's biological age? And like I said, this is a complex question to ask, but molecularly, where can we even start? We could look at genetics, epigenetics, transcriptome, proteome, metabolome, etc. But we were trying to figure out where should we even start thinking about this. Now, what has been previously done is when people talk about biological age, there have been some things that have already been tested. For example, telomere length. Most of you might be aware of this, and obviously telomere biology plays a very important role in lung aging as well as lung disease. In fact, a lot of IPF patients have mutations in telomere binding proteins. So clearly, telomere length seems to play some role in lung function as well. And so in the case of like aging biology, people were always interested in looking at telomere length and seeing if this could really predict biological age. Most of you know that telomeres are the end of chromosomes. And over time, as cells proliferate, these telomeres get shorter. And every time cells divide and the telomeres get shorter, at one point, at a critical telomere length, cells cannot proliferate anymore. And this is thought about as replicative senescence. And I won't go into replicative senescence a whole lot, but if you're interested, we can talk about it later. And so telomere length has long been thought of as a marker of aging. Now, a lot of the newer studies, however, when people look at telomere length, it does seem to have some correlation with chronological age, but does not seem to go really well with biological age. In addition, in addition, most of our cells are postmitotic, so they're not dividing, and so telomere length is probably not the best marker to look at. The other, the other groups have also looked at another clock or marker for biological age, and this is the epigenetic clock. Again, I'm not sure how aware you are, but I kind of just summarized it quickly in the slide. And the epigenetic clock looks at DNA methylation patterns. And based on at least the initial clocks were based on about 350 DNA methylation sites. And based on these sites, one could predict. Initially, they were all trained on chronological age. So the Howard's clock in 2013 was trained on chronological age. But since then, people have started to look at functional data, morbidities, diseases, etc., and use this data set as well, along with the epigenetic clock. However, one of the issues with the epigenetic clock has been we don't understand the molecular importance of the epigenetic clock yet, meaning because these marks are all over the genome, and in addition, every tissue has different epigenetic patterns, it's been tough to really decipher what does an epigenetic change mean to a phenotype. So for that reason, again, people that you can commercially buy these test kits really and get to know your epigenetic age, but it's not been clear what do these epigenetic changes really mean for the biology of aging. So when we kind of started thinking about this problem on a bigger scale, we went back to the central dogma. Everybody knows DNA makes RNA, RNA makes protein. But one of the most important components today is that metabolites basically are an additional layer that dictate all of these functions. And so also, metabolites are probably the ones that are much more closer to a phenotype. And because they can influence all of the other factors, including epigenetics and the transcriptome, we thought maybe looking at metabolites might be the most interesting. In addition, from what we know about interventions that could potentially work for healthy aging, such as intermittent fasting or calorie restriction, in all of these cases, again, it is metabolism that's heavily affected. So again, we thought maybe metabolites might be very important to determine one's biological age. In addition, my lab's always been interested in the role of cellular senescence, and I know our Dr. Yao is going to talk a lot more about this, so I won't go into it into detail. But as you might know, senescent cells accumulate with age, and they have multiple markers. But one of their key markers that distinguishes between a senescent cell, at least our current knowledge, is that what makes a senescent cell different from a quiescent cell, which also does not proliferate, is that it has a very particular type of secretome. So it secretes a number of cytokines, chemokines, metalloproteases, as well as lipids that we know of now, that affects neighboring cells and neighboring tissues, and this can drive both disease and aging. In fact, when I was a postdoc, I had worked initially with, I was at Scripps Research Institute and working with Mayo Clinic at that time. We had also found senolytics, which were drugs that could kill senescent or eliminate senescent cells. And if we did this in deceased or aged animals, then the animals started looking rejuvenated or at least biologically younger. Of course, now, there are many senolytics that are in ongoing trials. The last time I checked, about 15. I'm sure the number's changing on a daily basis. But clearly, senescence has a role in aging. If you ask me, again, is it a definite role, and is all senescence bad for you? That's a complicated question, and I think we are learning a lot more about senescence as we go. But clearly, does seem like senescence in multiple tissues accumulates with age, and removing senescence in an intermittent fashion might actually improve health. So again, when we really started thinking about how can we capture one's biological age, we thought integrating the metabolome as well as the SASP secretome, which is the senescent secretome, together might be able to give us a fingerprint for rapid agers compared to healthy agers. But even when we want to do this, where do we start? Should we look at a particular tissue? Should we look at blood, etc.? But we honestly landed up on blood for many reasons. One, this would be easily accessible. People are okay with doing blood tests all the time. You go for an annual checkup, and if you can get a little bit of your blood and do metabolomics and SASP secretome, that would be ideal. But in addition, there was also a biological reason for this. And that was a few years ago now. There were studies in the field of biology of aging called parabiosis, which now has been done in multiple disease backgrounds as well, where parabiosis was, taking blood from a young animal and then giving it to an old animal. And just this blood transfer was able to improve health of the older animal. So this suggests that something's in the blood that makes these animals younger and healthier. So we decided that probably blood was our first thing to look at. Now, I was really lucky that I had just come to University of Pittsburgh. And when I was recruited here, this was kind of a question I really wanted to address. And so I was very lucky because I'm in the Division of Geriatric Medicine and working with geriatricians who are very invested in our elderly population. And so they had just collected this cohort at that time where we kind of distributed them into two groups, healthy agers, where chronologically they were much older. They were at the age of 75 years or above. But biologically, they seem to do much better. And what I mean by that is that in a 6-minute walk test, they performed much better. They were able to climb up stairs. They were able to grip things, better. And so we had done all sorts of tests on these people, and they seemed to be healthy agers. Versus the other group was of 100 individuals were 65 years and above. And these people could not really do a 6-minute walk test without, stopping and inhaling and things like that, or taking some rest, sitting down, things like that. And clearly, they didn't have any major diseases. So that's an important point. But clearly, this group was not doing as well as our healthy agers. And why we looked at gait speed or the 6-minute walk test was gait speed seems to be, one is that gait speed is really dominated by multiple factors, including lung function, right? But also pain, muscle strength, etc. So it's a system that really integrates from multiple things and health from multiple tissues. And so that's kind of why we looked at gait speed. Also, in general, it's been known that a slower gait speed, even at the age of 45, could be a sign of accelerated aging. And so for that reason, we thought that gait speed was an important determinant of how one could be a healthy ager or a rapid ager. Now, in this group of 200 people, we collected multiple physiological parameters, and this paper is now published. So you can see all of the different parameters we have collected, and all this data set is also publicly available. If you need any more information, please reach out to me. My email's at the end of this talk, and I'll go through it at the end. And we collected serum from these people and did untargeted metabolomics and also looked at senescence secretome. So let me go through quickly, not through detail about everything we found in this paper, but like I said, it's been published now, and I'll be happy to answer questions. But the major findings from this paper was that we saw that the healthy agers were able to perform beta oxidation. So or basically break down their lipids in their mitochondria. So as most of you know, what is beta oxidation is when your lipids are broken down by your mitochondria, you need a lot more acylcarnitines. And this is what we saw in the healthy agers. Versus if your mitochondria are not functioning really well, lipids are then broken down in the endoplasmic reticulum through a process called omega oxidation. And omega oxidation, the byproduct of omega oxidation, is an increase in dicarboxylic acids, or DCAs, which is what we saw was higher in rapid agers. And just looking at how much acylcarnitines were present and how much DCAs were present in a person, we could really predict if one was a healthy ager or a rapid ager. Again, as you can see, healthy agers relied much more on beta oxidation compared to rapid agers. In addition, what we did was we looked at what metabolites could really predict a healthy ager from a rapid ager. And what we noticed was 25 metabolites in combination could predict a healthy ager from a rapid ager, and do this quite well. A lot of these metabolites are lipids, actually. And at the very end, I'll tell you a little bit of a snippet about what some of these metabolites could do. But for now, what we found in this paper was just combining 25 metabolites could predict if one was a healthy ager or a rapid ager. And this is what we called as the healthy aging metabolic index, or the HAM index. And as you can see, the HAM index was a better predictor of healthy aging compared to some of the other markers, including gait or comorbidities, cognitive scores, or frailty. Of course, the number one thing that's always asked and that you should do is you have obviously trained your model on a particular cohort. And so, of course, we wanted to see if the HAM index could still be predictive of healthy agers in a completely different cohort. And so we used a cohort called the RAP cohort from Madison. And again, in this particular cohort as well, our HAM index could predict healthy agers compared to rapid agers quite well. And as you can see, our HAM index was also quite good at predicting comorbidities some. But more importantly, because our training set had healthy agers who were chronologically older, one of the questions we got from the reviewers was, is this just predicting chronological age? Or is the HAM index really able to capture biological age? And so we looked at, the correlation between HAM index and chronological age. And as you can see, there was really no correlation. So we think the HAM index is quite a good predictor of biological age. We also looked, as I said previously, looked at senescence secretome. And we did notice that some of the SAS factors, such as MCP1 and IL-6, were reduced in healthy agers compared to rapid agers. And we have just started now looking at, what SAS factors are associated with what metabolites, because this could be very informative about both for interventions, but also tell us a lot more about biology. And interestingly, MCP1, which is a SAS factor that is elevated in rapid agers compared to healthy agers, was also correlated with a number of lipid metabolites, especially the ones that are DCAs, which come through the omega oxidation pathway. In the end, correlation studies are interesting, but my lab's really interested in understanding the what, why, and how. And so because we are so curious, we really wanted to see if we can do some sort of causal inferencing to figure out which metabolites could actually drive biological aging. And what we saw from our analysis were 3 metabolites were significantly could be putative drivers of biological aging. The first one, again, being a lipid. The second, which was beta-cryptoxanthine, again, which is positively associated with healthy aging. And beta-cryptoxanthine is a metabolite that's actually found in fruits and vegetables. So when you remember your mom telling you how you should have eaten your fruits and vegetables, maybe there is actually some science behind it. And in case of rapid agers, we saw an increase in prolyl hydroxyproline, which is a byproduct of collagen degradation to be highly elevated in rapid agers. Like I said, this part of the story has just been recently published. So you'll find more details there, but if you had any questions, I would be happy to answer them. But one of the things that, again, we found from this study was that the healthy agers seem to break down their fat in the mitochondria versus the rapid agers were not able to do that. And this was again just through the serum and the metabolites. So you wonder sometimes if really there is any correlation, and can you study this in something that's also accessible like PBMCs? So this was done in collaboration with Dr. Shruti Shiva, who is at the VMI at the University of Pittsburgh. And she had, from the very same cohort, the Solvit cohort that we had used for this HAM index, she had taken PBMCs. And when she looked at oxygen consumption rate through seahorse, she really saw no difference between the healthy agers or rapid agers. But what was interesting is if she looked at fatty acid oxidation, now it was clear that the healthy agers seemed to be, had increased fatty acid oxidation, beta oxidation in the mitochondria compared to rapid agers, which again correlates really well with our serum metabolite study. So this is very encouraging. And so we are starting to look at, what is the difference between beta oxidation and omega oxidation. Now, omega oxidation, where fat is broken down in the endoplasmic reticulum, now these DCAs are usually generated in our liver and in our kidney. So they can do omega oxidation compared to other tissues. Now, why would one do omega oxidation versus beta oxidation? One, at least what we know from other populations where there are mitochondrial diseases, these people do not have functional mitochondria, and then they seem to rely heavily on omega oxidation. So if you have sick mitochondria, one thought is then the lipids are broken down through omega oxidation. And this is seen, again, like I said, in multiple fatty acid oxidation disorders. But also, we know now that people also, there are people who consume large amounts of MCT oil, etc. And these, again, get broken down by omega oxidation to produce these DCAs, or dicarboxylic acids. And in all of these people who have these fatty acid oxidation disorders, they have high levels of DCAs. Of course, the question really is, is this DCAs in itself causing or driving biological aging, or is it the lack of the mitochondrial beta oxidation that's driving biological aging? And so that's kind of the questions we are getting into and trying to figure out mechanism now. But this is kind of what we are just starting to do. And I wanted to show you just a preliminary piece of data where if you took DCAs and just, put them on healthy growing cells, we saw that just two types of DCAs here, adipic acid and subic acid, the control cells seem really good. They grow really well. But soon after you give them these DCAs, we do either see senescence or we see an increase in cell death, both of which we know can be, both of which are hallmarks of aging clearly. So we are again looking at the mechanistic aspects of omega oxidation, which has not really been studied in the context of aging at the moment. So we're really excited that our initial metabolomics study has now given us some more insights on biology, and we are pursuing that further. With that, I want to, I hope I've not gone over time, and I want to give a shout out to my team. This majority of this work was really done by a postdoc, Shruti Himsanathan, who's fantastic and recently went to industry and is doing really well there. A lot of this work was done in conjunction with geriatricians, like I said. So Neil Resnick, Susan Greenspan, were the ones who really got this cohort together. And of course, my other collaborators. And of course, none of this work is possible without the funding that we have received. And with that, I'm happy to take any questions you have.
Thank you. Thank you so much, Aditi. Really interesting work, and I'm sure A lot of people are interested. So if you can just raise your hand and activate your camera if you have a question. In the meantime, maybe I can start. So I think looking in the blood for aging biomarkers is great, right? Because it's easily accessible. And you've shown beautifully that you can detect signals. So do you think that this will allow us to detect organ-specific aging signatures? Or do you think that there's no such thing as organ-specific aging anyways, and that's why this question is stupid?
Oh, no, that's a great question. And I do think that, our organs probably age at a different rate. I mean, there is no question about it, right? Can serum metabolomics tell us about organ aging? That is a great question, and we are in very early stages to answer that. However, things like, for example, now the DCAs that I talked about today had to come from one of two organs, right? From the kidney or from the liver. So in cases like that, maybe we can start thinking about where they're coming from. But of course, for all of the others, that it's going to be very complicated to answer it. I think, however, with the geroscience hypothesis in mind, knowing that if we can improve health in general, then we could delay the aging of multiple tissues probably at the same time. So I think that gives us some hope that even though our tissues might be aging at a different rate, we will still be able to understand the process of biological aging.
And the question is if we even have to care so much, if we can, as you said, right, if we think that they influence each other, right, then we want to interfere with the whole organism anyways, right?
Right, absolutely.
Yeah. All right, I see a question. Please, if you can switch on your microphone, Yati.
Yes. Hi, Aditi. Excellent talk. And it was very novel to see that there's a difference between like healthy agers and rapid agers. So my question is, did you check for any pulmonary function tests between these two cohorts? And then did you see any sex-dependent difference, especially in terms of the mitochondrial testing parameters?
Well, thank you so much, Niyati, for your question, and thank you for attending the talk. Yes, so pulmonary function. Sadly, we had not done a lot of pulmonary function, but now that I was, reading all of this stuff, I was like, how come we missed that? So I think from now on, again, if people in this group have other cohorts where pulmonary function's done, and either they have done metabolomics or would like to, maybe that's kind of where we should start working together. But your second question about gender and if that influences metabolites, I mean, absolutely it does. So we did see gender-specific metabolites as well. However, when we were looking at our Ham index, which is the metabolites that could predict rapid and healthy agers, these metabolites were independent of gender, as well as independent of even, smokers and non-smokers. And at least in our case, and how much, again, you have to take this data with a grain of salt, because we were not powered enough to do this. But it seemed to even, at least for African Americans and Caucasians that we had in this group, it seemed not to be biased between the two. Of course, we have to do a lot more studies with the bigger cohort numbers to really say that. But that is why we were highly excited that our Ham index, could, independent of gender, could even predict a healthy and rapid ager.
Okay, that's great. Thank you.
All right, so I think we can take one last quick question, please.
Hi, Dr. Gurkar. I hope I'm audible. It was a great presentation. My question is, were you able to correlate your Ham index to the epigenetic age at all? And I think you have touched upon it as well, but were there any correlations with the lifestyle like diet and race and things like that?
Yeah, thank you, Gagandeep. Thanks for the question. Yes, so epigenetic clocks, and this is kind of where our paper got stalled for many years at the reviewer's desk, because, you know, at that point, the epigenetic clock was sort of, and even now, in some ways, it is like the gold standard that people are using for biological age. One thing was we were looking at serum, so it was impossible to do the epigenetic clock. But we would love to do, you know, a new cohort and hopefully get some resources for this where we could really compare, or we could integrate, right? It could be very interesting because aging is so complex. I think it probably needs an integrative marker rather than just everything, you know, just an epigenetic or just a metabolomic or just a telomere length. If we could really do this in an integrated fashion to look at multiple things, I think that's what is going to tell us a lot more about aging. So we are very interested in doing epigenetic clocks in our future cohorts. But at the moment, we were not able to do that. And like I said, there are multiple metabolites that are influenced by a lot of things, like whatever we had about, we had very limited questions about diet, and especially, you know, it was a survey. So I don't think that can be really used to address diet questions. But things like gender, race, those are absolutely important. And at least our HAM index did not seem to care about these things. But in that paper, we have shown metabolites that seem to be associated with gender and with race. And that could be potentially, again, telling us a lot more about biology of aging in women versus men, and, you know, and again, tissue aging, etc. But this was not the HAM index. So the HAM index could predict independent of all of these factors.
All right.
Thank you.
Thank you so much. Super interesting. So we got very interested in metabolites and you got very interested in the lungs. So we all are very happy. Thank you so much.
Thank you.
Thank you so much, Aditi. Very nice presentation. Thank you, Mareike, for handling the question answered, for also responding to the chat. So we have a second speaker, and I would like to introduce Dr. Hongwei Yao. He is a professor in Brown University, cell molecular biology. Dr. Yao has a long-standing interest on senescence and biology of lung aging. So he has done his MD PhD from Hongwei University, and he joined University of Rochester, and then he moved for his faculty position at Brown University. So Dr. Yao he has achieved an R01 recently, and a couple of R01. He was stating that he's working on it, so look like he's very productive, and published in Nature Communication paper last year. So we would like to hear from Yao on lung aging with respect to postnatal lung injury, cellular senescence in lung development. So Dr. Yao, please go ahead and take over the podium, the microphone. You have 30 minutes, and then we'll have a question answer and the panelist discussion. Thank you.
Okay, thank you. Thank you, Irfan, for introducing. So it's my pleasure and honor to give the talk at the annual retreat of the non-aging working group. So my talk is about the cellular senescence in the postnatal lung injury and normal lung development. So cellular senescence was discovered 60 years ago by Dr. Herflick. So as we show here, it's young and old fibroblast. So the definition of the senescence is a term to use describe the cells that stop proliferation or divide and show the morphological change and with prolonged secretory activity. So as we know previously, we thought the senescence is kind of the cell arrest, which is not reversible. So however, accumulating evidence suggests that the senescence cell can re-enter the cell cycle for proliferation if the senescence maintaining mechanisms are not continuously provided. So this gives us many questions about the fate of the senescence cells. So such as how long the senescence can persist. So whether it's clear and how it's clear, and whether they can go through the second cell death, or whether it can resume in proliferation, right? There's many questions. Of course, we cannot answer now. So this is a review article summarize about the molecular feature as the Dr. Gurkar also mentioned about the molecular feature of the senescence cells. So usually the senescence is mediated by the two pathways, P53, P21, and P16. So the classical, the most commonly used marker of the senescence is the ACA beta gal. So of course, the mitochondria also can get damaged, as Dr. Aditi mentioned about the mitochondrial dysfunction. So once mitochondrial dysfunction is dysfunctional, can release the mitochondrial DNA, can trigger as damp. So of course, so many of the nuclear, the molecular features such as DNA damage, telomere shortening, and the NAMB1, the protein was reduced. So NAMB1 is kind of the nuclear envelope protein, which can be reduced in the senescence cells. And the senescence cell can secrete, can express unique surfactant. So this is really important for identify the senescence cells, or can be used as the therapeutic target for the remove or clear the senescence cells. So as we know, the senescence cell can secrete the SASP. This SASP can contribute to physiological functions such as wound healing and embryonic development, or the pathological changes such as aging and aging-related disease. So we are interested with the bronchopulmonary dysplasia, the BPD. So BPD is discovered by the Dr. Northway 60 years ago, so in Stanford University. So this disease is a chronic lung disease in premature infants, can caused by the mechanical ventilation and hyperoxia, oxygen supplementation. So BPD can affect 10 to 15,000 of the premature babies, so in the US. So lung pathological of the BPD is characterized by the alveolar and vascular simplification, which can persist into the adulthood. So of course, there are many of the pharmacological treatment. Although this treatment therapy can improve the survival of the premature infants, they have the minimal reduced BPD prevalence and associated lung injury. So therefore, a better understanding of the mechanism, can code or resolve new therapy to prevent lung injury in the BPD. So when we talk about the BPD, we have to understand the lung development, normal lung development. So you can see here why that we yes, we have to talk about we have to understand the normal lung development, because the infant born at the secular stage has a high risk for developing BPD. So in the human, as we know, the secular stage is started at the week 24 to the 36 weeks of the gestation. So after 36 weeks, they will become of the velar stages. So sheeps is the kind of the similar normal lung development, but just the precursors. So as the full gestation of the lung in the sheep, full gestation of the sheep is 150 days. So secular stage is the embryonic 110 and 140. So the mouse, the lung is kind of the really unique. So the secular stage can span from the embryonic 7.5 to the postnatal day 5. So therefore, the mouse expose the hyperoxia in the first days of the lives is the most commonly used model to the study human BPD. Of course, as I mentioned, larger animals can better model the clinical setting of the premature infant in the NICU. So this is just the lung morphology change. You can see here in the alveolar stage, the alveoli is well developed and really good for the gas exchange. So senescence is kind of the hot topic in overfield in the BPD. So there's a first paper published by Dr. Prakash from the Mayo Clinic. So his group used the subject and the control subject and the neonates who died within the 2 weeks with or without the oxygen supplementation. So they did the staining of the P21, P16, gamma H2X, that is the marker for cellular senescence, with co-localization with the alpha-synuclein. So they found the airway smooth muscle cells get sneezed after the oxygen supplementation. So as we know, the BPD is kind of small airway disease. So whether the alveoli have the senescence is not clear. And also the 10 years ago, there are two papers published in the field, kind of the back-to-back support the developmental senescence. So these used the SA-beta whole-mount staining to show the different organs, such as the neuron, such as the limb, such as the whiskey, such as the kidney. They show the cellular senescence in those organs. So they also found the developmental senescence is transient. They probably can clear by the immune cells, such as the macrophage, because they found the co-localization of the tunnel staining of the AC-beta suggesting clearance by the apoptosis. So here we don't know whether the senescence participated in normal lung development, and also whether the cellular senescence contribute lung injury with BPD remains unknown. So we hypothesize that the senescence participated in normal lung development, which is dysregulated and enhanced with the hyperoxy exposure leading to alveolar and vascular simplification. So to test this hypothesis, we first collaborated with Dr. Mareike Lehmann, who worked a woman infant in the Rhode Island. So we got the tissues from the premature infants, with or without the mechanical ventilation. So we did the LMB1 staining is the marker of the cellular senescence. We found here that the mechanical ventilation can cause, can increase loss of the nuclear LMB1, the staining, suggesting the cellular senescence. This is collaborated by the written paper published in the Real Journal from the Wisconsin group. So they did the P16 expression, the staining, and also nipple fusion staining. They found the senescence increased in the BPD lung tissue compared to the control group. So as we know, it's really, really difficult to have the BPD tissues, so therefore we collaborate with the Dr. Kurt Albertin, from the Utah. So his group has a long-term research interest in the BPD using a large animal, the preterm lambs. So as I mentioned earlier, the lambs, the gestation age is 150 days, 4 tons. So what they did, they used the pregnant lambs to get the surgery delivery at the 131 days. That is the secular stages. And those preterm lambs was subject to the mechanical ventilation for 21 days. We also did the LMB1 staining, and we did the gene expression of the P21. We found the mechanical ventilation can cause the cellular senescence in the lung. So larger animals always good, but very expensive. So I have the R1, which we used the 20 preterm lamb with cost of $1.5 million. So therefore we go back to the small animals, the neonatal mouse. So here we expose the neonatal mouse at the 95% oxygen for only 3 days, and then recovery until the postnatal day 60 or adulthood. So we did the different marker of the cellular senescence. So one of the two markers, SA-beta and LMB1 staining. What we found here, we found the neonatal hyperoxia can cause a transient increase of the senescence at the postnatal day 7. So that is one piece of the information. So another piece of the information is here. Compared to the PND3, the senescence was reduced at the PND7, PND10, and the PND60, suggesting the developmental senescence. Of course, this is under normoxia, without any of the oxygen exposure. Of course, we also did the different marker of the senescence, such as SASP, gene damage, and proliferation. So this also agreement with this paper, as I mentioned earlier, with the journal paper, they use the rats, they expose the newborn rat for the 70% oxygen for 10 days and wait until to the postnatal day 21. What they use a different marker, LMB1, nipple fusion, and the gamma H2X. So they also found the neonatal hyperoxia cause cellular senescence in the lung. So all this data suggests neonatal hyperoxia can cause the lung senescence. So the next question, as we know, the lung has tons of the cells, more than 40 and 50 types of the cells. So the question we want to ask, what is the cell specific of the senescence in the lung after the neonatal hyperoxia? So here we did the single-cell RNA-seq. So at the PND7, because this is the peak time for the cellular senescence after the little hyperrupture, we did the sort out the senescence cells based on the C12FDG staining. C12FDG is the substrate of the SA beta gal, which can tag the senescence cells. Then we did the single-cell RNA-seq and analyzes the data. So in combination of the single-cell RNA-seq and the immunofluorescence staining, we identified the senescence in the macrophage, type 2 cells, and the second quest myelofibroblast. So interestingly, we saw both the senescence cells at the macrophage. So therefore, we try to further characterize of this macrophage. Okay, so here we identified the 7th cluster of the macrophage. So some of the clusters are really interesting, such as the cluster 17. Okay, that you can see the number are significantly increased. Okay, so this is kind of the increased antioxidant, probably due to increased oxidative stress. And also the cluster 20 is would reduced by hyperrupture. It's kind of the reliever capacity would decreased by the hyperrupture. Okay, exposure. And also cluster 23 is kind of the doubled in the senescence cell, SD7. SD7, that means is senescence cells. We did the facts for the sort out the senescence cells. You can see the more of the polarization. So that's suggesting probably in the more M1, M2, the macrophages. So as we know, the lung, the macrophage has the residential alveolar macrophage and monocyte-derived alveolar macrophage and the interstitial macrophage. So we analyzed the data and we found these macrophages are all increased in the senescence macrophage. And we also, this senescence macrophage has increased M1, M2 macro, suggesting the pro or anti-inflammatory features if these cells are functional. Then we want to determine the mechanism of the senescence. So therefore, we used the culture of the MH cells in the alveolar macrophage. So we expose them to the hyperrupture for the 4 hours and 24 hours. So we did use a different marker, P21, LMB1, exclusion, and the gamma H2X. So altogether, we found the hyperrupture exposure increased cellular senescence at 24 hours, but not the 4 hours, indicating the time dependent. Then we, as mentioned by the Aditi, the SARS component is really important for the senescence cells have the paracrine effect on the neighbor cells. So therefore, we try to understand what the SARS component in the senescence macrophage. So here again, we use MHS cells exposed to hyperrupture. Then we sort out the senescence cells. So based on the DDAOG, there's another substrate of the AC beta gal. Then we put these cells overnight and connect the supernatant and did the proteomics. So interestingly, we found 2 groups of the pollutants. One group is uniquely increased in the senescence macrophage, which is not change in the lung senescence macrophage after hyperrupture. Okay, those of the pollutants mostly are the desmosome and the SAC kinase, tyrosine kinase, in tyrosine kinase, and histones such as the histone 3 and the histone 5, and the matrix pollutants. So another group of the pollutant is kind of the anti-inflammatory, and also is the pollutant can promote the repair, are significantly reduced, uniquely reduced compared to the lung senescence cell and the air group. So then we collaborated with the Dr. Wuzhen. So we did the proton analysis. It's kind of the tool to study the protein-protein interaction. Of course, you can see here, minimum of the protein can interact together, right? But these 2 proteins really interesting to us. So that's the SARC with the JUP, okay? SARC, as I mentioned earlier, that is the tyrosine protein kinase, okay? What is the JUP? JUP is the gamma catenate, is the component of the desmosome. What we found here, we found in the senescence macrophage, the interaction of these 2 proteins are significantly increased. Okay, previously, there's the paper to show the endogenous cell microvascular carry-on cycle-rich cargo can impair the adherent junction integrity. So now we are trying to understand whether this of the protein interaction sac and the tube can, from the senescence macrophage, can cause endothelial cells and epithelium the impair the endothelium and epithelium the adherent junction and the integrity, leading to the inflammation or the alveolar destruction. Then we want to see what the biological significance of the senescence the secretion from the senescence macrophage. To answer this question, we again use this supernatant from the senescence or non-senescent macrophage and inject them to the neonatal mouse at the PND7 and the PND10. Those mouse grew under the lomocule. Okay. So then a few days later, we did the number plot and did the staining of the VWF to show the visual numbers. What we found here, the secretion from the senescent macrophage can cause lung injury, okay, and also reduce the visual numbers. So however, there's no much significant change of the secretion from the non-senescent cells. So that is suggesting the clinical significance and the biological significance of the start from the senescent macrophage to the lung injury. So the second part is about the whether we can remove the senescent cells can protect against lung injury. So of course, this is the review article summarize a different of the strategy can target the senescent cells. So here we are interested with a few strategies. One is the senolytic, one is the xenomorphics, and the antibody drug conjugates. Okay, as mentioned by the Dr. Gurkar about the clinical trial of the quercetin derivative. Sorry, that's the announcement.
Uniform Whiskey 326. UW326, White Jeep. Please call.
Okay, that is a 70 clinical trial just updated from the 15th from the previous presentation. Of course, this is the senolytic can clinical trial in the aging, in the fibrosis, and in the Alzheimer's disease. So therefore, we also use this of the cocktails treated with the neonatal mouse at the PND4 to the PND14. Okay, so of course we found this treatment can reduce senescence markers and also can reduce the hyperoxia-induced lung injury, as indicated by the alveolar simplification and alveolar vascular simplification. Okay, so that is protect against hyperoxia-induced lung injury. So however, this senolytic cocktail, based on the few publications previously, this cocktail could have the toxic effect on the non-senescent cells. Okay, so I think it is really important to specifically target the senescent cells. So to use this effect, we try to develop the antibody drug conjugates. Okay, so as I mentioned earlier, this is a single-cell RNA-seq. We found the senescent cells, senescent macrophage have highly expressed of the UPA. So this is the plot, the gene is the plot. This gene can encode the UPA. So based on the previous publication, this of the UPA are increased in the neonatal mouse exposed to hyperoxia. If the genetic lockout of the UPA can protect against this lung injury. So this soluble UPA can also biomarker of the BPD in the premature infants. So then we want to ask whether the UPA positive cell are the macrophage are senesced. So we did the sort out the UPA negative and the positive macrophage from the neonatal mouse exposed to hyperoxia at the PND7. We found the UPA positive cells are highly increased of the P21, suggesting the UPA positive are senescent cells. So now I try collaborate with the company, try to develop the UPA antibody drug conjugates, so which is still is the underground. So yeah, we don't have any data to show this. So that's one thing. Another things, as I mentioned earlier, these are the skeleton SASP components. Okay, so many of these SASP components can regulate and interact with the P38. Of course, there's a decylative also can interact and modulate P38. So quercetin same thing can inhibit the senescence by the deregulate the P38. So therefore, what do? We treat with this senescence cell with the P38 inhibitor. Okay, then we connect the supernatant and inject the mouse again and measure the morphology. So what we found here, as we expected, the hyperduction, the senescence, the secretion from the senescence cell can increase the mean intercept, cause lung injury, and reduce vessel numbers, which was inhibited by the P38 inhibitor, okay, suggesting the P38 inhibitor could be the potential xenomorphic in treat the BPD. So that is the part of the lung injury. As I mentioned earlier, right, compared the PND3 and the senescence marker would be reduced at the PND7, PND10, and the PND60 under normoxia, suggesting the developmental senescence. Here we extend the embryonic stages. So what we found here, we found the senescence marker would increase in the PND0. Okay, that is the cellular stages. So we have so luckily, unless we want to answer what is the cell senescence, right? So luckily we have this paper published. No, that paper published in 2021 in the development. They have to show the single cell analysis of the mouse line development. They have the different time point of the embryonic stage of the lung tissue. They did the single cell RNA-seq, such as embryonic 12 to the postnatal day 3. So we analyzed this single cell RNA data. We identified the senescence here in the different cluster 2 and cluster 3 in the smooth muscle cell and myofibroblast. So now we are working what is of the function of the senescence cells in the normal lung development. So here we also try to reduce the senescence cells in the cellular stage. As we mentioned earlier, the cellular stage is the peak of the developmental senescence. So we did the treat with the dasatinib and the quercetin. Of course, they reduced the cellular senescence and also disrupted the lung development. So this suggesting reduce the cellular senescence in the cellular stage can disrupt the normal lung development. So here we want to summarize the lung senescence occurs in the BPD infants, mechanical ventilated proton lamps, and the hyperduction-induced neonatal mouse. So lung macrophage are major cells undergoing the senescence, which are heterogeneous. So unique the secretion from the senescent macrophage can cause the virulent vascular simplification. Senolytic and the senolytic and cephomorphic, so such as P38 inhibitor, can inhibit hyperduction-induced lung injury in the neonatal mouse. So the only programmed senescence, mainly in the mesenchymal cells, can orchestrate the normal lung development. So paradoxically, what the importance of the timing of the senescence embedded at cellular immobility also hyperduction-induced alveolar simplification. So this is kind of the schematic to show the normal developmental senescence is really important for proper normal, the alveolarization. So once we export the hyperduction, this can cause lung injury. So that's important for timing. When we treat the BPD, with the drug, we don't want to remove the normal lung developmental senescence cells. So here I want to thank the lab members, particularly the lab Phyllis Dennery and her lab member, because she mentored me for many years, 8 years, and collaborated with her lab members still on this paper and the grant submission. Of course, I want to thank the Cobri and the collaborators and the funding support. So, okay, I want to stop here, and if you have any question, so we can discuss. Thank you.
Great. Thank you, Yao. Wonderful presentation and keeping on time. So I would like to ask Mareike to take over, please.
Yeah, thank you so much. Beautiful presentation. So do we have any questions already from the audience? You can also type something in the chat if you want to. So in the meantime, I know we have one. Yes, please.
So Hongwei, this is Ganesh Kunduri from Milwaukee. Wonderful talk and very intriguing data on the dual roles of senescence in both lung development and in hyperoxia injury. So the question that I have is, if you take hyperoxia exposed macrophages and you're putting them in the lung, in a normoxia exposed lung, and it's inducing alveolar and vascular changes. So if I imagine the alveolar macrophage being adjacent to the epithelial cell, and if hyperoxia exposed macrophage is affecting the epithelial cell, so is it affecting primarily type 1 or type 2 cell? And if it is type 2 cell, is it more on the progenitor role, or is it on the surfactant function? Have you looked at those details?
Yeah, that's a great question because here we use the in vitro culture, for that. But in the vivo, we found that the senescence cell majorly is a type 2 cell, even it's a small component. It's only less than, I think, the 8%. Okay, that is a mouse model. But from your group, you saw the different of the senescence in the different cells, epithelial cells, endothelial cells, right? I think probably that is the kind of the difference of the exposure, the duration, such that we only have the only 3 days, only in the secular stage exposure. But for you guys have the 10 days exposure, which we span from the secular and the alveolar stages. And also, yes, about the probably the rat and mouse have difference. So that is, yeah, that is we are working on that. Yeah. Thank you, Dr. Condor. Yeah.
All right, then I can ask my question. So I have a more technical question and a question to your antibodies. So when we sort cells based on senescence-associated beta-galactosidase activity, we always get a lot of unspecific signal in the macrophages, or what we think is unspecific signal. So I was just wondering how you deal with this in your single cell experiment.
Yeah, so that is really good because as we know, the macrophage also has the larger lysozyme, right? Has the increased the SA beta gal. So each cell without the cellular senescence. So that is here we present here is kind of the in vitro study, and now we are try to what do we try to? We try to use the in vivo model. We try to clear the senescence macrophage in the vivo. What we have this, we collaborate with the person, with Dr. Xu Min in the University of Connecticut. So they have the P21 highly expressed the cells. So we try to use this mouse to cross with macrophage, the Cree mouse, the Flox mouse. We try to remove the P21 highly expressed of the cells and see what the change of the morphology. Yeah.
All right. Yeah,
Thank
you.
you.
And if I may, another question to the antibody that you have. So it's the UPAR antibody. So what kind of functionality will it have? So do you think that this will induce apoptosis in the end in the macrophages, or what? So do you just want to mark the macrophages with this, or will it inhibit something?
Yeah, that is we just beginning this of the kind of the project. What we doing now, we use this antibody to conjugate with the dye. First of
With the
all,
dye.
first of all, we want to see whether this antibody can be internalized by the cells. Right, so that is the first phase. But of course, it's really, really important when we treat with the antibody and whether we change the cell function, such as apoptosis, such as what the proliferation. Right? Yes.
So this would then also go into the line of the CAR T cell work on UPAR.
Yeah. Right.
Yeah. Okay. Very interesting. Thank you so much.
Thank you.
Do you have other questions? Irfan, Beata, anything from your side?
No, it's a great presentation, Yao. So this is very intriguing about the lung development and senescence. So when you analyze the, just minor question. So when you analyze the single cell data, the published lung map, did you see any senescence on those senescence markers, senescence genes on cell types on those data?
So that is either in the injury phase or in the developmental phase?
No. This is a single lung map data.
Oh, lung map data.
Yeah, the already published data, you said fibroblasts and smooth muscle cell you are targeting. So do you see any those data on different lung developmental stages?
Yeah,
lung map data. Did you see any senescence over there in terms of?
Yeah, that's a good question. We have to ask the Jocelyn. So she is a computation biologist. So we want to ask her to look at the lab data, whether it can correlate with our finding. Yeah, that's good suggestion. Thank you, Ivan. Yeah.
Yeah, good.
I have a question to this. I mean, it's really intriguing that you see this the homeostatic senescence or the developmental senescence you see at different stages than the pathological senescence, the hypoxia-induced senescence. So is it only a matter of timing, or do you also see really qualitative differences in the type of senescence that you see?
Yeah, it's not just the timing, also it's about the cell specific. Yeah.
So it's about the specificity of the cell, and do you think it's also the senescence will be different, or is the cell type that's affected different? Do you know what I mean?
Yeah, that is a complicated question. I think, yeah, it's really hard to answer. But if we use the kind of the technology such as, the reporter mouse, so if we have the kind of the reporter mouse, what we can do, we can, as I mentioned earlier, what is the fate of the senescence cells, such as the developmentally, we saw the senescence in the early stages, right? So whether these of the cells can steer what's going on in after the hyperrupture, right? So that's is a really good question, but we need to answer, use different of the technology to answer that. Yeah.
So Yao is the same extension of Mareika's question. You said that whether it is, what she's asking is a qualitative or something really happening. So same question in a different way. When you said that in those published papers, the cells are senesced in different areas of the body or that organ, and then you also mentioned there is a clearance of senesced cells by macrophages, there potentially by macrophages.
Yeah.
Would you like to elaborate that? That will answer Mareika's question too, whether you see some clearance by macrophages on those early developmental senescence, or is just an artifact, or is just a qualitative thing what happening in programming?
Yeah, so that's we don't know in the developmental phase, but we have that data to show. So okay, even the macrophage can engulf the senescence, the type 2 senescence cells. So we showed there. So that you can see so many of the problems when we started the macrophage senescence, because you don't know whether it's real, is senescence in the macrophage, or is engulf of the senescence, other cells, other type of the senescence cells. Yeah. Yeah. So that's why we have to use the genetic approach. We can, what we can do, we can probably can delete use the P16 or P21 highly expressed cells in the macrophage and see what the change of the
Or in other words, you can do that. You can have the myeloid cell knockout mice crossing with senescence during early development and see whether if you don't have macrophages, then what happens to especially interstitial macro. Yeah, that's kind of.
That also is a good approach. Yes.
Okay, thank you. So, Mareika, more questions from the audience rather than from the panelists?
I have one more question. So actually, it was a great talk. Thank you for sharing your data. You mentioned that you observed DNA damage. I wonder whether you had a chance to look whether this may be related or maybe not to impaired DNA damage repair. And if there is any if yes, if there is any potential to identify any factor which can play a role in therapeutic strategies.
Yeah, so we have, that's a good question, but we don't know what the repair protein is changed, in the DNA repair change, but it's a good suggestion for us to look at. So we also saw I didn't show here, we also saw the increase of the oxidative stress. So this oxidative stress probably can, of course, impair the damage the DNA, also probably can cause the compensatory of the increase of the repair proteins. So which we are not know, but it's a good suggestion for us for future study. Yeah.
All right. Thank you. I think it's super interesting, and it links very nicely to what we discussed with Aditi to look at organ-specific senescence. We also need to look at cell type-specific senescence. So I think there's a lot that we still can learn, right?
Yeah, and also look for metabolomes.
Yeah, no, absolutely, yes.
Yeah, in terms of the metabolomics, we also have some of the data to show in the macrophage cellular senescence. So we found increase of the glucolysis. When we inhibit of the glucolysis, can inhibit the hyperglycemia-induced senescence. So we wrapped this data and tried to submit, to JCI, and then they'll see how it goes. Just recently, yeah.
Excellent.
All right. Thank you. Then Irfan, I give over to you.
Yeah, congratulations to all of you and very good job done. And this is a successful mission here for the retreat. So we spoke 2 webinars. We heard 2 webinars and we saw our panelists. And so we still would like to put this slide to show you that we are here for you. This is from the ATS members. If you are members, if you are not members, please do join the ATS and click the button on RCMB. I don't know how it works, but definitely we are part of the RCMB. And if you want, you can have critical care as well. So we are here for promoting lung aging research and therapeutics, as well as any new thing comes. For example, when we had the retreat actually last year, this year ATS. So what we had, our members came and they gave really good suggestions. So this is from the members. So Mareike compiled these topics. So these topics, we would like to ask you to nominate speakers. If you would like to present yourself, of course, please do email us, one of us, and we would like to organize another retreat very soon in the spring or in summer or after the ATS or definitely in the fall. So increase the awareness in basic biology, lung aging, and omics of aging, for example, epigenetic clocks. That's what we heard today. And repurposing senolytics to prevent lung aging. Then anything lung mapping of what we heard today from Dr. Yao. Would like to see how we can link with the aging. And then anything precision medicine based on senolytics, xenomorphics, antibody-based conjugate CAR T cells, or anything you have which you would like to pursue in this, of course. And then the most important thing, I still feel that omics of aging, which you have all the transcriptomics and proteomic, metabolomic data combined to come forward for the lung development and lung diseases. So with that, I think the time is up. But any other thing, I would like to give 3 minutes for the members. If you'd like to tell us something, your feedback, and if not, then we'd like to conclude this webinar or the retreat. With that, I would like to give my microphone to Beata and Mareike to conclude this. Beata, please go ahead.
All right. Yeah. I mean, if there's nothing else, then thank you very much also from my side. I think this was very exciting, and I'm looking forward to the next time.
Thank you for joining us.
Okay, so I think with that, we end this. Thank you so much, okay?
Thank you. Bye.
Thank you. Bye-bye.
All right. Bye. See you soon, Beata.